I'm going to explain the effects of norepinephrine and epinephrine on a pacemaker cell. Throughout the pictures, I use different colored candies and pellets to help explain how it all works together. In my pictures, the neon green pellets represent the layers of the membrane of the cell. The blue sweet tart represents the hormone, which in this case is norepinephrine or epinephrine. The hormone is binding to the red jelly rancher, which is a beta-1 adrenergic receptor. This receptor only will accept norepinephrine and epinephrine. This process is metabotropic, which means it is done through a second messenger. The three jelly bellies make up the G protein, which will later signal adenylyl cyclase. The G protein is made up of three subunits, alpha, beta, and gamma. The gamma subunit is represented with the green jelly belly, beta with pink, and alpha with light blue. Before the hormone binds to the beta-1 receptor, there is a guanosine diphosphate, or GDP, attached to the alpha subunit, which is represented with the orange pellet. When the hormone binds to the beta-1 receptor, it adds a phosphate, which is called phosphorylation. The GDP then becomes guanosine triphosphate, or GDP. The subunits leave the beta-1 receptor, and then the alpha subunit leaves the other two so that it can later bind elsewhere. The alpha subunit binds to adenylyl cyclase, which produces cyclic AMP when ATP is present. The enzyme that catalyzes this reaction is phosphodiesterase. The adenylyl cyclase is represented with orange starbursts and the cyclic AMP with the purple sweet tart. The cyclic AMP then goes and binds to the target protein, which in this case is protein kinase A. The protein kinase A is represented by the red starbursts. When cyclic AMP binds to protein kinase A, it allows the calcium ion channels to open up more than normal. When looking at a normal action potential graph of a pacemaker cell, also known as the sinoatrial node, you see that sodium ion channels slowly open at the beginning, causing depolarization. This segment is called funny ion channels, or the funny current. The funny current goes from resting potential to threshold. Once it reaches threshold, the calcium ion channels open and cause further depolarization. The graph looks the exact same when it is affected by norepinephrine and epinephrine, except the calcium ion channels open faster due to the adrenaline, which causes a quicker depolarization. The whole process of the ion channels opening and closing will happen more frequently when affected by norepinephrine and epinephrine rather than at rest.